Hi Ninja Nerds, in this video we are going to talk about the digestion and the absorption of carbohydrates. So let's go ahead and get started. So let's imagine for a second we take a scenario where we're going to ingest a particular food substance that's rich in carbohydrates. And for example here, I love pizza. Pizza is awesome. It's amazing. It's delicious. And what we're going to say a part of the pizza is like the crust, specifically the crust and the bread part of the pizza is going to be the part that's rich in carbohydrates. But now the question you should ask yourself is, out of this, what are the most significant parts? What are the actual carbohydrate molecules that are in that pizza that we want to digest? There's a bunch of them, but we're only going to primarily focus on two significant types. One here is this big one here. And this big molecule here is called amylopectin. And we'll talk about this one in a second. And the other one is this linear molecule called amylose. There is another one that we can digest from substances that are like fruits and veggies, and that's called cellulose. And there's other ones like chitin and lignans, but again, we're primarily going to focus on the ones that are actually going to be absorbed. Cellulose is actually not able to be broken down and absorbed because we don't have the enzyme cellulase. So because of that, cellulose, which is a part of the different plants and veggies, really it's just a part of our dietary fiber. And so that helps with increasing the stool size and helping with easing the actual bowel movements. And there's also been other health benefits too of it, that they say that it can control the bacteria, metabolism, like the prebiotic mechanisms, and they can play a role in insulin, controlling an insulin release. All right, anyway, getting back to this, let's say that we ingest this piece of pizza. And in the piece of pizza, there's rich amounts of amylopectin and amylose. What I want you to realize about this diagram is that this big brown tube here, we're going to represent as our GI tract. But we're going to go through parts of the GI tract where the different digestive enzymes are going to be found. That's why we have these little tubes here. So if you imagine this tube is going to be specific to like the oral cavity. So let's say that this tube is really specific to the oral cavity. Okay. And this is actually going to be the next one, which is going to be respect with the pancreas. Now the pancreas makes different types of enzymes like pancreatic amylase, and that gets released into a specific part of the GI tract around the area of the duodenum. Okay? And then the last part here is we're going to zoom in on these enterocytes. So these are going to be, this is one gargantuous enterocyte with a bunch of these different brush border enzymes. And this is the part where we'll focus on different types of disaccharide enzymes, as well as a couple other different types of ones here. And then we'll talk about the absorption of these monosaccharides and then the movement via the hepatic portal system to the liver. All right, so let's go ahead and get started. So the first enzyme that we want to focus on here is going to be secreted by this gland. And this gland here is going to be our salivary glands. There's a bunch of these bad boys. So salivary glands, you have extrinsic and you have the intrinsic salivary glands. Like the parotid is an extrinsic, or the submandibular or the sublingual, and then there's the intrinsic like the labial, the palatine, the buccal glands. But they're making different types of saliva. Now the saliva, mainly from the extrinsic salivary glands, is rich in a particular enzyme, and that enzyme is going to help in the chemical digestion of this amylopectin and amylose. What is that chemical right here? That chemical that we're going to focus on here is going to be what's called salivary alpha amylase. And realize there is another name for it, just in case you read it. It's also called thialin. Thialin. Get the A there and then the L-I-N. Okay, so these are the different, this is the special enzyme that we're actually going to release out here into this part of the oral cavity. Now let's say here's the tube. This is going to be like that duct that we're going to release it in if it's via the extrinsic salivary glands. Now once the salivary enzyme, salivary amylase is out here, let's treat him like a little Pac-Man. I like a little Pac-Man little guy. This is our salivary amylase. Now this salivary amylase, he is hungry and he is going to come and start digesting certain types of chemical bonds on these carbohydrate molecules. But in order for us to really understand this, what I want you to do is just very, very briefly, this is not a biochemistry video, I need to very, very briefly talk about just a little bit about the chemical structure of these monosaccharides, or together these polysaccharides. So let's come down here for just a second so we can get a tiny little view here on the one of these monosaccharides. So if you look at a monosaccharide, it's a 6-carbon sugar in this situation. And I'm going to show you where those carbons are so that we have some understanding of what these bonds are, because that's the important point. So here is going to be this monosaccharide. Now on this monosaccharide, we have 6 special carbons. The carbon that's close to the oxygen here, that is carbon number 1. Come down here, start just doing a circle, go clockwise. This is carbon 2, carbon 3, carbon 4, carbon 5, and here is going to be carbon 6. There's 2 types of bonds that we are going to see in these different types of carbohydrate molecules. Okay, and what are some of these bonds? We're going to see one type of bond that's going to be between the 4 carbon of this guy and the 1 carbon of another glucose molecule. So for example, let's say I take this glucose, or monosaccharide, it should be specific, and have it bonded to another guy over here. So now I'm going to have this. This is usually a hydroxyl group. Now, same thing here. This is the 4 carbon of this monosaccharide. This is the 1 carbon of that monosaccharide. And then again, 2, 3, 4, 5, 6. This bond right here between the 1 carbon of this guy and the 4 carbon of this guy, it's a special type of bond. What is the name of this bond? This bond is called a alpha 1 to 4 glycosidic bond. That is the name of this bond. Okay? Then we have another one. Let's say I bring one more monosaccharide out here. One more out here, and I'm going to go here, here, oxygen. And again, put my CH2. And then my OH group here, but H is going to leave here in a second. Same thing here. 1, 2, 3, 4, 5, 6. There's another bond that we can do. This next bond is I'm going to attach another glucose unit onto this part here. So now you actually do keep the oxygen here, and then you get another molecule. And then look, right here is the 6 carbon here. So now, if you remember, this is the 6 carbon of this monosaccharide. That is the, it's next to the oxygen. That's the 1 carbon. This is the 2 carbon. This is the 3 carbon. That's the 4 carbon, 5 carbon, 6 carbon. Now this new bond that we just formed, the new bond is between the 1 carbon of this monosaccharide and the 6 carbon of this monosaccharide. So now, what is this bond that is between these 2 molecules? That bond right there, this one right here between the 1 and the 6, let me kind of circle it like this. This is called a alpha 1 to 6 glycosidic bond. And the purpose of the alpha 1,6 glycosidic bond is to allow for the branching of these different types of polysaccharides. So now we should understand a little bit, a tiny, tiny bit necessary for us to understand this actual structure here and what type of bonds this enzyme is going to break. So now that we understand these bonds, what I want you to realize is that amylopectin, as you can see here, look what kind of bonds he has. Well, he has this part that are linear, going from 1 to 4. So he has a bunch of alpha 1,4 glycosidic bonds. So he has 2 different types of bonds here. He has alpha 1 to 4 glycosidic bonds. And he has these branching points right here and right here. Those are his alpha 1 to 6 glycosidic bonds. Now, that is the difference between amylopectin and amylose, because what can you see here with amylose? amylose only has 1,4 glycosidic bonds. So amylose only is containing alpha 1,2,4 glycosidic bonds. All right, sweet deal. What salivary amylase does is he only targets the 1,4. So what he's going to do is he's going to come over here and he's going to try to break this bond. He's going to come over here and he's going to try to break this bond and this bond and this bond and this bond and this bond. So he is going to start destroying some of these things. So he's going to start breaking the alpha 1,4 glycosidic bonds. Now, when he does that, when he breaks this amylopectin, and he's also going to break this bond over here. So here, I'll bring the little Pac-Man dude down here too. There's my little Pac-Man guy, my little salivary amylase. He is going to break, again, these alpha 1,4 glycosidic bonds. Now, when he breaks the alpha 1,4 glycosidic bonds, he's going to break the majority of this amylopectin and the majority of this amylose into another different type of molecules, these 3 special types of molecules. We're going to represent here, one is called maltose. So one of the molecules that we can get a lot of is we can get what's called maltose. Another one that we can get a lot of is what's called Malto Triose. So another one here is going to be what's called Malto Triose. So this is Malto Triose. And then we get one more, and the big difference with this one is that he has to have those branching points. And if you remember those branching points, what was specific? It had to have those alpha 1,6 glycosidic bonds. So off of here, I'm going to have a couple branching points. Here, let's just do it like this. And then one more here. So now I have this molecule, and this molecule is called my alpha limit dextrins. So my alpha limit dextrins. Okay, so here's what I got. I have my Malto Triose from the breakdown here. I have my maltose, and I have my alpha limit dextrins. We're going to refer to these molecules, primarily Malto Triose and alpha limit dextrins. Usually when you have 3 or more different monosaccharides, we call them oligosaccharides. And if you only have 2 of these monosaccharides, you refer to as disaccharides. All right, so the next thing that's really interesting is the salivary amylase. We know it's made by the salivary glands and pushed into the oral cavity. But not all of this carbohydrate digestion is occurring just in the oral cavity. That's the thing. Food isn't really in our oral cavity that long. For me, it's definitely not, because I'm swollen them suckers whole. All right? So that's important to understand, that this chemical digestion of carbohydrates isn't really just happening only in the oral cavity. It's actually happening in all the organs on the way to the stomach. So if you think about it, when we swallow the food via deglutition, it moves down through what other structures past the oral cavity? It moves past what's called the pharynx, and then it can move into the esophagus, and then it moves into the stomach. Now, you have to remember that the stomach is super acidic, and salivary amylase, his enzymatic activity is optimal within an acidic range, but not super acidic as in the stomach. So it doesn't really last very long in the stomach, but it does allow for some chemical digestion to take place. I want you to remember, though, the salivary amylase doesn't account for a super large amount of the actual chemical digestion. Maybe 10 to 15% of the actual chemical digestion of polysaccharides is occurring by the action of salivary amylase. The next enzyme is also going to be contributing a decent amount here to the breakdown of amylopectin and amylose. And what is this molecule? So if we go to the pancreas, we talked about these molecules specifically when we talked about the intestinal phase of gastric secretion. But if you remember, we had like a ballooned out area here, and this ballooned out area was consisting, it was called the acini. And the acini, you had your acinar cells. And the acinar cells made a bunch of different types of digestive enzymes. One of them was called pancreatic amylase. So now, pancreatic amylase, salivary amylase, be thankful. They pretty much do the exact same thing. So now we're going to secrete the pancreatic amylase into the duodenum, because you know really what happens is you have the gallbladder, and the gallbladder has what's called the common bile duct, and the common bile duct comes down and fuses with the main pancreatic duct and forms the hepatopancreatic ampulla. And then what happens is, based upon a hormone, you know there's a hormone called cholecystokinin. He acts on what's called the hepatopancreatic sphincter, or the sphincter of Oddi, and triggers the relaxation of that muscle to lead to the ejection of this pancreatic amylase. Not only that, but a beautiful thing here is CCK is so nice. He says, let me help you out also too. I'm going to stimulate the pancreatic acini to make pancreatic amylase also. But again, remember, what does pancreatic amylase do? Pancreatic amylase, look, we're going to draw him like this. Here's this guy. This guy is going to do the same thing. We're going to represent it here in this orange. He is going to cut specifically only 1 to 4 glycosidic bonds. When he breaks the alpha-1,4 glycosidic bonds, he's just going to increase the digestion of amylopectin and amylose, so here, into maltose, Malto Triose, and alpha-limit dextrins. So that is the function of this little cool little enzyme, and this is called pancreatic amylase, this little dude here, and then this maroon guy here is our salivary amylase. So beautiful thing, beautiful thing. All right, now we go down here to where we're still in we can be in the duodenum, the jejunum, the ileum, and so pretty much the entire intestine, because enterocyte is going to be for your intestines, primarily that of the small intestine. So let me actually put that. This is an enterocyte, but primarily of the small intestine. A lot of the absorption occurs primarily in the small intestine. Very, very little things that are absorbed in the large intestine, mainly water and electrolytes. Okay, so now we have a bunch of different enzymes. Here's where we get to these guys. You see these guys, they're literally bordering this actual enterocyte. These enzymes are referred to as brush border enzymes. So let me write this over here. We have what's called brush border enzymes. And there is of 4 types that we are going to discuss. One is going to be lactase. Another one is going to be maltase. Another name for it is called Gluco Amylase. Another one is going to be sucrase. And there's another one which is called isomaltase. But what you're going to see very interestingly is that sucrase and isomaltase are actually complex together as one big enzyme, but made up of 2 individual components, which is pretty cool. Now, one more thing about the brush border enzymes is, I didn't represent it here, but when you look at enterocytes, your small intestine cells, they actually have these small little cytoplasmic extensions. And these little small cytoplasmic extensions are really important for being able to increase the surface area, not just for chemical digestion, but also for absorption. What are these little cytoplasmic extensions called? They call them microvilli. And what's so cool is, I didn't represent it here, but this microvilli, these enzymes, I'm going to represent here is blue. That's the lactase enzyme that we're going to talk about. And then over here, we'll do red. That's going to be the next one, which is going to be the maltase enzyme. And then after that, we can have the sucrase and the isomaltase enzyme. And again, these are also going to be over here too. So the whole point that I'm trying to get across here is that the microvilli are the ones that contain a high concentration of these brush border enzymes. And the whole purpose of the microvilli, to remember that, is to remember that it increases the surface area for chemical digestion and absorption. So for digestion and absorption. So that's pretty cool. There's other structures in the intestines that also increase the surface area for digestion absorption, like the plicae circularis. And another one is the intestines. We'll talk about this when we talk about the histology of the GI tract, but generally, when you look at your intestines, they have like this type of structure. They come around like this, and what happens is the enterocytes are actually covering these things, and that's where you get those little microvilli here. But these little hills, where the actual enterocytes are sitting on, these hills are called villi. Okay, so these are called your intestinal villi. Okay, that one is just a villus, but you have a bunch of these, which they're plural. It's called a villi, your intestinal villi. So you have intestinal villi, you have microvilli, and another thing called plicae circularis, or circular folds, and they all help to increase the surface area for digestion and absorption in the small intestine. All right, cool. So now what we're going to do is, these are the big things that you're going to get released here. All right, this maltose, Malto Triose, alpha limidextrins, but there's other things that can be released here too. These are just one of the significant ones. Another thing that can be released here is what's called lactose. Okay, lactose. And lactose is just a disaccharide. It's a two glucose and galactose unit, basically. And what happens is, here I'm going to put GLU for glucose and GAL for galactose. These are the two components of the lactose molecule. The lactose molecule is going to run into this blue enzyme. And what is this blue enzyme called? This blue enzyme is called lactase. Some people might know about this enzyme because you might not have it. Sometimes in genetic situations, you don't express this enzyme, or maybe due to environmental stimuli, you actually inhibit the production of this enzyme. And if you don't have this enzyme, guess what you can't break down? You can't break down lactose. So you can't eat any type of substances that have lactose in it, which is commonly substances with like dairy, so milk and cheese and different stuff like that. Because it can cause severe diarrhea, cramping. You'll be, tooting a lot too. So that's significant to know a little bit about that. So here's what happens. The glucose and galactose molecule, the lactose together, they come in and they fit into this little active site of the enzyme. So look, here I'm going to put it like this. So here's boom, 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 boom. Here's that guy, and we're going to bring it together here like this. Now, what happens is the lactase is going to break that bond. It's going to break the glycosidic bond between glucose and galactose. Once you do that, guess what you do? You break this bond here. By breaking that bond right there, it is going to separate these two guys into their individual components. And what are these individual components? We're going to represent them as circles now. This is going to be glucose, and this is going to be our galactose. So we'll put GLU and GAL here. All right, cool. That's an important thing to know. That is the job of lactase. It breaks down lactose. If you break down lactose, you break it into two components, and it is going to be glucose and galactose. All right, sweet, that's it. Now we come to the next enzyme, this red enzyme. This red enzyme is a very special enzyme. He is called maltase. And again, another name for maltase, I'll write it over here, is called Gluco Amylase. That's another name for maltase, is Gluco Amylase. What it can do is it can take maltose, right? So it can take maltose. It can take Malto Triose, and it can break these puppies down. Now, here's the thing. Maltose is actually going to be made up of two glucose units. So now here's what I'm going to do. I'm going to put again in here another little thingy here. Here's a little guy here, and here's another guy right here. I'm just doing the maltose here, but I realize that Malto Triose will have pretty much the same thing here. And what I'm going to do is if maltose or Malto Triose fits in here, guess what's going to happen? Maltose is going to be made up of two glucose units. So when you break the bonds, the glycosidic bond between maltose, it's going to break this off into two components. One component is going to be the glucose, and the other component is going to be the glucose. So it's such a cool thing there. So maltose is 2 glucose units, whereas lactose is a glucose and galactose. Now we come into this next one, this double enzyme, if you will. This enzyme, we said that there are 2 parts. So here's what we need to do here. The pink part is called sucrase, but they're combined together. This part here is called isomaltase. Here's what is cool about these enzymes. Sucrase, it can take on maltose, it can take on Malto Triose, it can take on alpha limit dextrins. But what it does is it also functions in breaking down another molecule, and that other molecule is called sucrose. So now let me put in here a sucrose molecule. So here's one. I'm going to bring this over like this. Okay, now this molecule here, this right here is called sucrose. But again, it can break down. It can break down maltose, it can break down Malto Triose, and it can even break down a little bit of the alpha limit dextrins. But the alpha limit dextrins is primarily for this enzyme, and that's what we'll talk about here. But now, sucrase is really going to love to digest this sucrose. If it digests the sucrose, it's going to break the sucrose down into 2 components. And what are these 2 components that we're going to get out of here? So one component that we'll get out of the sucrose is going to be glucose. The other component is going to be fructose. Oh, man. So now, as a super fast recap, lactase breaks down lactose into glucose galactose. Maltase breaks down maltose into 2 glucose molecules. And sucrase breaks down sucrose into glucose and fructose. But realize that sucrase can also act on maltose and Malto Triose. The last one here is the isomaltase. Like I told you, the isomaltase is very, very specific. He loves to break down alpha limit dextrins. So now what this isomaltase does is he's really good at breaking down the alpha limit dextrin. So here, let me put this here so I have more room here in this active site here of this enzyme. Now, what I'm going to do is I'm going to take here, and I'm going to have a glucose molecule, or, a monosaccharide. I'm going to have them like this. And again, remember, you have that 6 carbon popping up here. And then coming off of this, I'm going to have my glucose units right here, or my monosaccharide units. Now remember, we said that this bond is the special bond right here. These 2 bonds. These 2 bonds right here are the alpha 1,6 glycosidic bonds. Isomaltase is one of the special enzymes that primarily targets the alpha 1,6 glycosidic bonds. So now look at this. We're going to kind of fit this like that. Yeah. And then we'll have another part of this enzyme like this. So now we'll kind of bring the enzyme like this a little bit. Yeah. All right. So now if you look at it, what is this going to do? The whole purpose of this is I'm going to break the alpha 1,6 glycosidic bonds. By breaking this bond, the alpha 1,6 glycosidic bonds, it breaks down the alpha 1,6 glycosidic bonds, which helps all these other different enzymes, maltase, lactase, sucrase, to continue to break the 1,4 glycosidic bonds. So that is what is so cool about this guy, is that whenever this reaction happens, it breaks down these alpha limit dextrins. And when it breaks it down, it gets rid of those alpha 1,6 glycosidic bonds. And as a result, it might release out of here some glucose. It might even release out of here some other molecules too, but we're going to primarily say here that it's going to be glucose, because that's the main monosaccharide that we want to absorb. All right, so now lactase broke down lactose, glucose galactose. Maltase broke down maltose into 2 glucose units. Sucrase broke down sucrose into glucose and fructose. But the isomaltase was that really special one, and the reason why it was really special is because it was breaking the alpha 1,6. It was breaking alpha 1 to 6 glycosidic bonds. And remember that as it is breaking that alpha-1,6 glycosidic bonds, that again is going to help all of these other enzymes to continue to break down these oligosaccharides or disaccharides into their individual monosaccharide units. So now we are pretty much ready to absorb these puppies. Here's the cool thing. There's two main mechanisms that we're going to get all of these sugar units in. Fructose is a really cool guy because fructose says, you know what, I don't want to go with glucose and galactose. I want to go on my own. So here I'm going to have this pink guy here, and this is going to represent our fructose. Fructose has a special channel that will facilitate his diffusion from the intestinal chyme in this case, or the lumen of the small intestine, into this enterocyte. Do you know what this protein that is going to help in his facilitated diffusion is called? This protein is called GLUT5. So it's a glucose transporter type 5, and it's going to transport the fructose from the intestinal lumen into the enterocyte. Oh, that's cool. All right. So that's going to be by facilitated diffusion. The other guys aren't so lucky. They're going to be going against their concentration gradient, glucose and galactose. So here's the thing. On every cell in your entire body, you have these special protein molecules. These protein molecules are constantly pumping 3 sodium out of the cell and pumping 2 potassium ions into the cell. And this process is requiring the utilization of ATP into ADP in an inorganic phosphate, right? Because it's utilizing energy. This is an example of primary active transport. We're pushing the sodium against its concentration gradient and pushing the potassium against its concentration gradient. But by doing that, pushing the sodium out, what happens to the concentration of sodium inside the cell? It's going to decrease. Well, guess what? It decreases so much that the sodium concentration in the actual intestinal lumen is much, much higher than the concentration of the sodium inside of the cell. So sodium says, hey, glucose, hey, galactose, you guys can follow me, and if you follow me, I can get you guys into the cell because I've already helped out in this process. So sodium comes into the cell, and as a result, glucose and galactose will follow. So here I'm going to have glucose, and here I'm also going to have galactose. And again, what molecule did we bring in here? We brought in sodium. And guess what we can do with that sodium? We can shut it out again through that sodium potassium ATPase transporters. So this right here is through an SGLT transporter. They call this transporter the SGLT, sodium glucose transporter. That's literally what they call it. They call it a sodium glucose cotransporter. And that's pumping the sodium into the cell and pumping the glucose and galactose into the cell. So it's a symporter technically, right? But again, the reason why it's doing that is because sodium is able to move down its concentration gradient and bring glucose and galactose with it via what's called secondary active transport. Now here's the cool thing. We have fructose, we have glucose, and we have galactose. These guys right here are our main monosaccharides. So glucose, galactose, and fructose. These guys right here are our main monosaccharides. This is the big ones that we want. Here's the cool thing. On the basolateral membrane, the part where we're actually going to have the blood, right? So this is the lumen side. This is the actual basolateral side where the actual blood vessels will be. Right here, you're going to have these blue transporters. And these blue transporters are really cool. These blue transporters are actually a GLUT transporter, but primarily GLUT2. So these right here are called GLUT2 transporters. This one right here and this one right here. The GLUT2 transporters are nice because they're not too specific to just glucose. They also can take on fructose. They also can take on galactose, and they can also take up glucose. So now we're going to have here, boop, and we're going to have here, boop. So from here, what am I going to have coming out here and going into the actual blood? I'm going to have glucose, I'm going to have galactose, and eventually coming up underneath it here, I'm even going to have fructose. What will happen is these guys will get absorbed into the actual intestinal veins here. So there's some actual capillaries, and what will happen is these guys will get taken up by the intestinal capillaries on the vein side of it, and they'll actually get absorbed and taken up eventually through different types of veins. You know, there's the superior mesenteric vein, there's the inferior mesenteric vein. Here, we'll write all this down here. So what happens is you have a bunch of different veins. We're going to call this vein as collectively, we're going to call it the hepatic portal vein. But there's a lot of different structures that are going to be incorporated into this. We'll have another video when we talk about the liver lobules. But we are having a bunch of different structures like the superior mesenteric vein, the inferior mesenteric vein, the gastric veins, the splenic veins, and even the pancreatic veins, which is important because we need insulin to help in this process here. So all of these guys are coming together to form the hepatic portal vein, and what the hepatic portal vein is going to do is it's going to take this glucose and it's going to take that, it's going to take the glucose, it's going to take the galactose, and it's going to take the fructose and perform certain types of metabolic activities. Primarily, since we're feeding, we're going to do what's called glycogenesis. We're going to do other processes like maybe even some glycolysis. So that is going to be important, and we'll talk about, again, this in more detail when we talk about the liver lobule. All right, engineers, so I hope all of this made sense. I hope that you guys really did enjoy it. If you guys did, I'm begging you, please hit that like button, comment down in the comment section, and please subscribe. Also, if you guys get a chance, please go check out our Facebook, our Instagram, and please, even our Patreon account. If you guys have the opportunity to donate, even if it's $1, it would make the difference in us being able to make the most high quality and best videos for you guys' enjoyment. So please, if you guys can, go check that out because we would truly appreciate it. As always, Ninja Nerds, though, until next time.